Hello, everybody. And as usual, because it's the weekend, we're here with Scooby. But because it's the weekend, we're going to listen to a little Stacy.
He's innocent. The day always comes if you're innocent.
Get on the train before it leaves the station. Wisconsin versus Avery, a complete fabrication. Get on the train before it leaves town. The truth train is coming, gonna run them down, run them down.
Do your YouTube, Scoop.
And we all know Stephen and Brendan were blamed, but it does not take a brain surgeon to see that they were framed. And yes, we know what this was about. The civil suit, the settlement that did not want to pay out, want to pay out.
Yeah.
And you may hear this song, oh, maybe you don't get it. I suggest you look up TikTok, manage to walk on Reddit. There you will find trial transcripts, evidence files, and you will start to fathom the corruption within the court. And as you shake your head because everything seems defended, you will find yourself in company of 3 unbanked judges at the sentence. But don't you worry, you're not going insane. You're just having to sit down on Zelda's truth train. Or
it leaves us calm.
Final destination. The full acceleration. Train. Before it leaves town, the truth train is coming, gonna run them down, run them down. Get on the train.
Yeah, there we go. All right, so that's.
I don't know who I'm singing with.
Yeah,
I'm singing with the wrong one.
So everybody, make sure you tune in for that too. That's a big deal as well. And we're here today with Scooby. I know everybody's been long awaiting the return of Scooby, and here she is, everybody. So we're happy to have her back. How you doing, Scoobs?
Good. Okay. Oh, I have to turn the sound off on this YouTube, right?
You need to turn it. You need to mute.
I need to mute. Okay. Now I'm right. Yeah.
There you
okay.
Go.
Oh, no wonder. I was singing. I don't know. I was all confused. Okay. I was.
No worries.
Yeah, it was kind of like, what did Rule say? He's a musician, a canon. Yeah, because there was one it was delayed. There was one, so I didn't know which tune to sing to. So I just did a little of both. But I'll have to work on that.
Alrighty. Okay, so everybody's Linda K says Scooby. All right, Scooby Dooby Doo. Falcon Apata says Scooby Dooby Doo. Hi, Scooby.
Hi.
So as you guys know, we got some interesting happenings in the case recently. We all know about the state's response recently, obviously, where they basically said that they still have not violated statute.
Yeah.
Because the bones that were returned to the hall box were possibly human. That's what they were. So.
Sorry? They say you a copy of that. It's going live.
You need a minute?
Oh no, my assistant Lois is just asking me some questions about what this is going on. My friend Lois is with me, so yeah, right. She still is trying to understand this whole concept. She thinks it's so that's okay. Carry on.
So we know that the state said that, which is essentially they're acknowledging the possibility that what they gave the hall box wasn't Teresa. I mean, at the very least, that's what we can determine from that response. They don't know what they gave the victim's family. Okay? So I think that's interesting. The way that KZ puts it in her reply is brilliant, where she basically says that now Gon and Fallon are arguing that that they didn't violate statute because basically they gave the Hallbachs, they reawakened the Hallbach family's grief in 2011 to give them animal bones. So it's very interesting stuff, folks. So let's see what's going on in the comments here real quick. Linda K says, love the red, Scoob.
Yes, that's so. My friend Lois just asked me, is this trying to bring the truth out? And I said, exactly. That's exactly what we're trying to do.
Right. Depending on the lighting, it can look green, blue, or red. So no, I think it's a red wall, right?
Yes, it is. It's a rose. It's actually a rose-colored red wall.
Right.
Mm-hmm.
Everybody thought it was a green screen.
To the colorblind, it's a green screen.
So, we're going to obviously, one of the reasons why we were getting Scooby on here is that there was a lot of people wanting to hear mtDNA explained a bit. So that's potentially one of the things we're going to do here today. We're going to talk a little bit at one point here about something that Scooby's been looking into recently. Something kind of, that she's been digging into, and I think it's pretty darn interesting, actually. It just shows a lot more of the kind of dishonest little doings. The state, just more things that make the state look sketchy. And I think it's interesting. So, and it's all with the bones again. See, it's funny how there's so much sketchiness around these bones. Why is that? You can't help but ask or wonder. And we're going to try to get to some of that today. And then, basically just talk about, the same stuff we would normally talk about. The bones are going to probably be a big part of what we talk about today. Obviously, Zellner's reply to the state's unsolicited response. We'll be probably bouncing back and forth today, but, we're going to be talking about all those things. So, James Crane says those guys are between a rock and a hard place. I'd say that's about right. Lloyd Bass says, hashtag follow them bones. Hashtag BOL.
Follow them bones.
Hey, Lloyd, hashtag BOL.
The bones hold a lot of answers.
The bones. Let's see here. The state needs to get their S together if they're going to lie. If they're going to lie so much, right? They got to at least get a straight story, right? Between them. Maybe they need to huddle up. Maybe that's what the state was going to. Or maybe they don't. Maybe we don't let them huddle up, and then just continue to watch the disingenuosity abound, right? They're definitely not. They're definitely not all on the same page, and they're all starting to step on each other, in my opinion. So.
Well, it's a mess. And when I first started looking at the DNA reports, I thought, well, this is going to be real simple. And then the next layer of complexity became, well, I don't really know what's being tested. And or where it came from. And the issue with the bones was that clearly the objective was to link Stephen Avery, link the bone and I, fragment, these are bone fragments or whatever, because they use vague language. They talk about charred remains and tissue and
right.
In various reports, there's various terminologies being used. But what seemed clear to me was that and in the response, he made a big boo-boo because it was not what was testified to in trial. In trial, they made it seems to me that they wanted to give the perception, which we know is very important to Ken Kretz, that what was identified as belonging to Teresa Halbach came from Stephen Avery's burn pit.
Right.
However, now that there's more information available recently, it doesn't appear that that's the case. So did Leslie Eisenberg not review her notes the night before the trial? Or did she intentionally claim, say that she sent things to the FBI that she didn't?
Right.
It is not for me to accuse, but something to certainly, I think about.
Mm-hmm. Yeah, certainly. Everybody's saying hello, Scooby. Scooby and Erekose, yay. So we're, I guess we'll start with mitochondrial DNA.
Okay.
So help everybody, for instance, understand where the mitochondrial DNA is coming from. Essentially, it's obviously another part of a cell of a human cell, besides the nucleus, which is where normally they would get the DNA from or get the readings from, right? So it's just to help people understand it better. What's the mitochondria? Why does it last or somehow hold on and stay viable for testing longer than the rest of the cell, if you can, for everybody?
Well, this is great because this case, that's what makes this case so fascinating, is because the mitochondrial DNA is really critical. And I don't think a lot of people necessarily understand. And I don't mean that derogatorily. I just mean if you didn't have study this, and really in the 90s did people really even start talking about mitochondrial DNA, when it started to be looked at in diseases. And mitochondria are my passion, so I'd love to talk about them. So who knows? Okay, who knows that you have two genomes? I'm just trying to get a feel for the audience. Yes, press 1 if yes you do, or 2 if no, you had no idea you had two different genomes.
You're asking everybody.
We're going to have class here.
Okay, so you're asking everybody, say that one more time.
Yep. Press 1 if you know you have two genomes, and 2 if you didn't know.
A lot of 2s.
Right. Okay, so this helps me know where to start. Mm-hmm. Nobody else wants to admit. That's okay. Thank you, people. Very brave. Okay. Well, you do. You have two genomes. When we talk about DNA, we're normally talking about, look what I have. Okay. Okay, we see it in my paper. Yep. Okay.
Pretty well.
So this is a cell. Can we see? Okay. In your cell, you have the nucleus. Okay. In the nucleus, you have one copy of your nuclear DNA. And when normally you're talking about DNA, this is your molecular blueprint. This is the DNA that is the blueprint for everybody that you are. And when we talk about DNA, this is normally what's being talked about. Now, every cell except what cells? Who knows what cells don't have DNA? Anybody? Anybody? One from mom, one from dad, right, Lloyd Bass? That's how it works with the nuclear DNA. So this is sometimes called, this is present in every cell except, I'll give the answer. Red blood cells.
Red blood cells?
Red blood cells, okay? So when you're taking a swab of blood, blood that might look nice and red isn't necessarily, doesn't mean it's going to be any better source of DNA than blood that doesn't look as red, because it's the white blood cells where you get the DNA from.
Right. And if you have enough red, you got white in there, right?
Hopefully. But what else has a lot of white blood cells? And this makes it a good source. Anybody? If you've been in Scooby school, which needs to be, my Scooby people will know this. There's one other fluid that has a lot of white blood cells.
Everybody was answering sweat. No, it's red blood cells, folks. Somebody
yes, Russell, spit.
Yeah?
Spit. Your saliva has in fact, per volume, your saliva has as many white blood cells as blood. So saliva is an excellent source of DNA. Hence, when Sherry was, splaying her spittle over her tubes, that was a very significant source of contamination. Just a little aside there.
Right.
So some cells are better sources of DNA than others. Okay, getting back. So everything other. So you have one copy of your nuclear DNA per cell, except in your red blood cells. Now, you have these little organelles. Did anybody, I'm going to date myself, but remember, did anybody have the big plastic model of a cell, like in biology class? I have to move back. But you have little organelles, lots of little organelles in your cell. From 100 to 1,000 mitochondria, abbreviated MT, per cell, depending on the type of cell. What do mitochondria do? Does anybody know?
They're your powerhouse for your cells. They create energy, right?
Yes, they create. Look at my picture. Look at my picture. People will laugh. Ziggy laughing pretty gosh darn hard. Okay, well, I didn't say this was professional, but it's hard to do this. Okay, so yes, ATP. ATP is what fuels everything that you do. So without your mitochondria, you could not exist. Without a lot of things, you could not exist. But mitochondria produce your ATP. So you've got 100 to 1,000 mitochondria per cell. Now, each mitochondria has its own genome. And you know why? This is, I'm sure you're all just very curious, but
I am actually.
you are? Okay. So the hypothesis is that way back in time, mitochondria were independent living little bacteria. And so somehow, they produced, they were really good at producing energy. And so our eukaryotic cells, or just, other kind of cells, decided these little bacteria came into the cell, and we decided, oh, they are of benefit to us because they can help us produce energy, ATP. And so they just sort of coalesced. And then as they absorbed, a lot of their DNA got absorbed into our nuclear genome, but the mitochondria still retain their own genome. So now the thing about the mitochondrial genome, our nuclear DNA has about 3 billion base pairs. Okay, when we're talking about base pairs, I look like this will be art. I could sell this. We could sell this on your for money. Here, Scooby. This is like Scooby originals. Art. This is a cell. Okay, so DNA. Okay, exactly. Truth Monster is all exactly A, T, C, G. It's just a combination. It's like computer. Code is all 1 and 2s. DNA is all A, T, C, G, A, T, C, G, A, T, C, G. Well, your nuclear, genome is in, 3 billion of those. The mitochondrial genome, there's some things about it that are different. It's a circular because it's like a bacterial, because it's thought to basically have come from originated from a bacteria. It's bacterial-like. So it's a circle. It's double-stranded, like our DNA, but it's a circle. It only has about 16K base pair. So it's a much smaller genome.
Right.
Okay, so the reason mitochondrial DNA can be useful now, of course, technology has improved, but in cases where, especially in burnt cases where tissues are burnt, which damages the DNA, the mitochondrial DNA is a little protected. First of all, you have a lot more of it. So per cell, you're going to get 1 copy of nuclear DNA. Sometimes they say two because they split it in half, but whatever the semantics, it's 1 copy. But you might have
1,000 mitochondrial.
Right. So you're just going to have a bigger volume of DNA.
Right.
Also, the mitochondria is enclosed in this double thick double membrane. So it's very protected. And then it's felt that the circular structure of the mitochondrial DNA helps, whereas our nuclear DNA is arranged in chromosomes, which have ends. So it's a little more protected for that reason. But basically, it's just the sheer volume of it. It's the sheer volume of it and the fact that it's a little hardier, being of bacterial origin. So per cell, if you can't get enough nuclear DNA, often in cases where remains are very badly decomposed or burnt, especially. Now, burning is the worst case scenario. So even if just because the heat is very damaging to the DNA. So you can find like a body that's been submerged in water and it might look all bloated and gross, but the bones are still, in pretty good condition to get DNA from. But burning, the heat really damages the DNA. And then when the heat is bad enough, as in this case where the bones were calcined, we heard that term calcined being used. That means essentially that all of the organic or material has been burnt away. So bones are composed of basically two. There's the organic, which is the carbon, carbon, oxygen, hydrogen, organic molecules, which the DNA is considered part of. And then bone also, and we'll get to this later on when we talk about these particles. Bone also has some minerals, minerals, calcium, phosphorus. Most people are familiar with those things, as well as some other minerals that make up their structure. And that's what you see, when you cremate someone.
Yeah. And he asks, is the white cell count higher if when somebody is sick or has an infection?
Yes. Okay. Normally, because that's the white blood cell's job, is to, if you have a significant infection, that's the white blood cell's job, is to fight. That's what they do. They fight infections. So normally that's one of the, often if you have an infection, they'll test for a bad infection. That's sometimes how it's picked up. So now we're getting off. So anyway, so that's okay. So the reason, so that's why today we're starting to, so when I talk about an STR profile, do you guys know what that means? 1 for yes or 2 for no? Nobody's in.
Okay, 1, got a 2.
2. Okay, yeah. 2. See, this is helpful because sometimes, you just start spitting out these terminologies, Eric, you know what I mean? Mm-hmm. And you just assume people understand, but maybe they don't. And that's why, I don't want to. So this is great because, I don't want to just be talking. No, Ziggy, you're not dumb. You just don't, you just don't have the background. And that's, and if people are just using these terminologies, but they're never defined, then it's sort of useless. So let's take the time here to define them so we all know what we're talking about. Exactly, Rule. We're all learning. That's why we're here. I would have no purpose. I would have no reason. I would have nothing to do tonight if I didn't have you guys to teach.
Mm-hmm. So I know it's a DNA, it's a form of DNA testing, a method of DNA testing.
Well,
obviously you're going to give us a little deeper.
Well, not really, because we're not going to, we can do, that would be like a whole video, but just so STR stands for short tandem repeats.
Right.
Because that's going to be, okay. But when you...
Naturally left-handed?
Yes, I'm left-handed. Short tandem repeats. Okay, so whenever you see STR testing, that's referring to the nuclear DNA, or we'll say nDNA, okay? So that's the kind of testing that they do in the crime labs. Okay, that's a kind of testing that Sherry Culhane did. And, again, that might be beyond the scope of this video, but we can have many future teaching videos if people are interested.
Yeah. Well, we'll continue.
We'll see.
As often as they ask for clarification on DNA stuff, we'll do another.
Or they might say, "Oh, good Lord, please." Stop. Oh, yeah, I forgot. I'm trying not to laugh because I broke a tooth. I was eating trail mix. I was trying to be healthy. See this guy? And I broke a tooth on an almond and it cracked. So I look kind of like a hillbilly. So I'm trying not to laugh too loud. Okay.
It's a good thing I don't like almonds, but yeah, those things are hard.
Don't make me laugh. Yeah.
And those things are hard.
They are. It's very dangerous. I was better off. I should have picked the M&Ms out like Lois. That's what Lois does, right? she picks out all the M&Ms. And I'm left with these hard nuts. Okay. So we can't tell Scoop. Thank you, Ziggy. Yeah, I tried to, but I'm self-conscious about it. So anyway, but okay, but who cares? So let's go back. So we won't go into, because that would be too long. That would take up, the whole time, but that could be another video. But STR, short tandem repeats, is the kind of testing that Sherry Culhane is doing. And what they're with the nuclear DNA. So what they do, you see those numbers in those reports, like there's a 12, they'll have a loci and an 8, for instance. Because, right, like Lloyd said, one comes from the mom and one comes from the dad.
Mm-hmm.
So what those numbers are, what those short tandem repeats are, so obviously you've got 3 billion base pairs, in your nuclear DNA. And we can't, here, let's just say hi to Scooby quick. Mm-hmm. Can you turn around? Can you see Scooby?
No.
Oh. I thought he would get picked up on the camera. I had to turn.
Scoobs, what's up, Scooby? Oh, I'm in your ear.
Oh,
there he goes. Hey, what's up there, buddy?
Oh, I can't. Am I getting? Oh, he's not showing. Scooby, I gotta get Scooby.
He is.
There he is.
What's up, dude?
Scooby's yawning.
He's like, I'm tired.
Yeah. Okay. He's listening. He's listening. He is here though. I just had to interrupt for a quick and let people, because I knew people would be asking about him. So he is here. Okay. Okay. Anyway, back to our subject. So when you see those, so just when you see that term STR, when you hear about loci and see like numbers like that, you're talking about nuclear DNA. Now, what they do, so a short tandem repeat, usually therefore, so somebody discovered that there's 3 billion base pairs in your nuclear genome. So obviously they're not going to sequence the whole thing, but somebody, discovered there's certain regions of DNA that are variable, where we all have sort of the same pattern, but we're all a little bit different. Mm-hmm. And the way that we're different is in the numbers of these repeats. So short tandem repeats, so it's like maybe a pattern, like they're all different, they have different names. You've all seen the profiles, mm-hmm. and those are named. So one might be AGAG. Okay, so AGAG is the repeat. So if it's repeated 12 times, you're a 12. If it's repeated, 20 times, you're a 20. If you're, blah, et cetera, et cetera. Right. So that's what that means. That's what those numbers mean.
And that is a sequence that reoccurs often, right? So, and how many that you have is the indicator that they look for, right? Got it.
Exactly. And the loci is the...
And there are many other things like AGAG. There's, obviously, right? 3 billion others, you said?
Well, there's 3 billion of these things, but these particular locations,
right,
they're called a loci, and you'll see that they have numbers, or they have names like D2,
oh, right.
Okay, that's
the certain spots on the DNA
on the that tells you which chromosome it's on and where it's located. So the difference and the reason, these are just areas that used to be called junk DNA. In other words, they don't code for anything, but they're useful in forensics because they can differentiate one person from another. Now, I don't even want to get into this, too much into this, but this is still a young science. We don't know a lot about the inheritance about these, and I won't even get into the statistics tonight.
Right.
But there is controversy. Okay, I'll just say that. So now we couldn't, so let's say this just as an obvious example. So the reason we have to use these non-coding, so your DNA can be divided into two regions, coding and non-coding. Coding is the part that codes for something, a gene. So it codes for your eye color, or that makes your kidney cells, or that, makes...
To code would mean it's inactive. It's active.
It's right? Active, right? It makes a protein. It makes something. So obviously, those things need to be very specific. They need to have the correct pattern of letters, because if there was if they were different,
we'd all be very different, right?
Well, you might not work.
Right.
So, for instance, if your aromatase inhibitor enzyme didn't work right, you'd be messed up. So a little change there. So it's not, those aren't good places to tell people apart, because we all have genes. All of our genes have to be pretty much the same. And that was the usefulness of these short tandem repeats, because there's a lot of variation in people. Right. And so when they're doing, because somebody, let's say, so I'm taking out, so they know where these, let's say here's a chromosome. I'm not going to write out all the letters, but here's a chromosome. It's like A, G, all the numbers are in between. So they're able to pick out using this thing called primers. So I know right here, and I can use, there's a way of doing this.
Lift it up a little.
There's a way of doing this. I'm sorry. I'm getting, yeah, I have to get used to the camera. So there's a way of doing this. So I can go, so what they do during this testing is they go out and they find the location where this, let's say I'm looking for my D2 loci. Loci is just a location. That's where this thing is located. So I have two little things called primers that are going to pick out that piece. And in that piece, I'm going to have that's where I'm going to have maybe 8 repeats of my sequence. They're all different for each one. But again, I'm just let's say my sequence is AG. Oh, and notice in the test Sherry does, Ken Kratz says this in his email, there were 2 that are called penta. Yes, I'll let somebody in the smart crowd say, how many are there letters are in the penta repeats? I'm trying to get audience interaction here.
Say it again.
How many? There's 2 in the test Sherry used. There's 16 loci, and 2 of the loci are referred to as penta something. How many letters are in those repeats? Mm-hmm. That's just a little trivia question. Penta.
Russell says 0.
No. How many letters are in the penta? Usually they're 4. Classically they're 4, but there's 2 that are called penta. 5. Yes, truth monster. Penta means 5, right? Quad is 4. Right. So the penta D or the 2 penta ones have 5. That's all that means. They just have 5 letters in the repeats. But anyway, that's a little trivia. But the bottom line is that what these tests do is they're not sequencing. They're picking out this chunk, and then they run it. So the primers go in, and they pick out the chunk that they're supposed to. Mm-hmm. So you've got your chunk that's picked out. And then they run it. Well, in the old days, they ran it on a gel, but now it gets run through a very small tube using electricity. And based on its size, because obviously, if there's 4 repeats, it's going to be this big. If there's 8, it's going to be this big. Here, let me get, if there's 20, it's going to be this big. Okay. Mm-hmm. So they're going to be different sizes. Mm-hmm. So they're going to basically, because DNA, deoxyribonucleic acid. This is getting too sciencey, right? Deoxyribonucleic, it's an acid, so it's charged. So these are going to separate out by size. So basically, I'm going to know, and then that's going to be and that's going to be come out on a graph. I'm going to know whether I'm a 12, 20, or 4 by the size of the chunk. Okay? Nobody looks at the actual sequence in there and sequences those, it's just assumed. Okay, that's basic STR profiling.
Right.
Okay,
right.
Now the difference, and this is why mitochondrial DNA sequencing takes much longer, is because we're actually going to sequence the genome because it's much smaller. So in the mitochondrial genome, because it's such a small genome, it's what's called highly conserved. It only has 13 genes, believe it or not, that code, and they all code for proteins in the respiratory, your respiratory complex, not your breathing. That's the complex, the electron transport chain. And I could talk about that all night. The electron transport chain is what takes what you eat and converts it in this incredible reaction and converts it into ATP by breaking a water molecule in half. And so you only have 13 genes. So the rest.
Well, we'll save that very interesting conversation for another one, but obviously.
For never. Yeah, I'm sure people are saying.
Actually, I'm actually kind of intrigued, but we got enough to get through tonight. So.
Okay. So HV1 and HV2. Okay. Well, this is not to scale. This is okay. HV1 and HV2 are two regions of the mitochondrial genome that we don't really code for anything. They're just sort of like they're analogous to the STR regions of the nuclear DNA. So they are they're basically the same. So these are regions. So in other words, if you have a mutation in one of these 13 genes, then you're in trouble. But you can have a mutation in the hypervariable areas, and it just doesn't really mean a whole lot. Right. And so what they do when they do a mitochondrial DNA analysis, and that's why it takes so long, is that they actually take these hypervariable regions, and I don't know, I don't remember offhand. They take part of them. They take about 300 base pairs, and they actually sequence them. So they don't just pick out. So they actually, and that's a little more labor intensive. So they actually get the readout: AT, 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 G, C, D, T, A, T. All 300 of those. Okay. Right. When they do a mitochondrial DNA, so they're actually sequencing it. So they are. So that's a difference in the testing. So you won't hear and maybe when we another day or night we can go through the results of the mitochondrial DNA test and to see how they're reported. So what they do when they're giving the results is that they compare. There's one mitochondrial genome that is the standard, and basically they compare how the genome that they've analyzed deviates from this standard genome.
Okay.
And anyway, so mitochondrial DNA is sequenced, and so it is named by how it.
So I have a question.
So that's kind of an.
In order to match that up, do you have to have an mtDNA sample to match that up too, or
yes.
Okay, so because you couldn't match that up against, say, an STR sample.
No, they're completely.
It's just two different things, right?
Two different things,
right? And if I may here for a second, to be clear, what we're actually testing in an mtDNA is we're testing the actual DNA strand of a kind of a foreign, bacteria that lives in our body and helps us. So it's actually it's actually like of another being, essentially.
No.
Not a being, but another.
No. It's
not.
you. No, that's going.
It's not, so it's the same DNA.
it's not the same DNA, but it's you. Okay. I was going back into, that's how they think it evolved, that at one time it was a free living bacteria that came and now became part of you. But no, it is your DNA. It is you. It is you. It is not a foreign being.
But it doesn't look like,
it's a
but it doesn't look like the DNA in your nucleus.
No, it's different.
It's different. So that's whether or not, okay, so wherever I was getting wrong there, my point is it's different than STR. And that in and of itself means then that you have to get specifically mtDNA. So that's why they had to go to her mother, correct? Right? They had to get DNA from, they had to get mtDNA sample from Karen.
Well, that's an interesting point. And they did that, but they didn't have to. Now, mitochondrial DNA was traditionally used because now STR tests are getting more sensitive. So they're able to use them now, but still with burned bones, you know, especially back at the time of this case, the only way you were going to get anything. And notice Sherry's sample didn't come from bone, and that's a whole nother story. But the only way that you were gonna get something from burned bone was with mitochondrial DNA back at the time. And nowadays, still, that's basically, they're starting to be able to get STR profiles, you know, which is what the guy is doing with the Andy testing. Right. But that's only very recently, and it's still hard. But I lost my headphones. What was the question again?
I was basically saying, I was just pointing out how the mtDNA is a completely different DNA strand compared to our normal DNA strand. It's actually like a separate DNA strand within us that is now a second way. It's like having two ways to identify because it's actually different than our actual DNA strand. So that's the part that I'm learning here and finding interesting. Then, then I was asking, where did they get what did they get to match up that this was Teresa's DNA? What were they matching it up with?
Okay, now, and this is another question that I had, or I don't know if this is why the FBI does this, because ideally, okay, now, and then the inheritance is different, which we didn't get to, but ideally, you would match, okay, if I had an unknown sample, so my bone is my unknown sample or my remain, well, I would want to get a sample from Teresa to match that up to. And traditionally, I know what I was going to say, so because it was so hard to get nuclear DNA from badly burned and badly, you know, charred remains, mitochondrial DNA has been used a lot by the military in identifying soldiers' remains and victims of disasters, and actually AFTO, the military lab, is very good. They're actually a very good mitochondrial DNA lab because they've done a lot of it and a lot of work and a lot of research, all blind testing with mitochondrial DNA testing. Right. So the mitochondrial DNA could be used when there was no other way, when there was no other way to get the other way. Now, normally, when you're
obviously they didn't have Teresa, though, right?
Obviously, ideally, you would match something. So you would match something from the person, okay, because that's the most accurate, right? So if you know, but sometimes, like in cases of if someone was gone for a long time, they might not have anything left. So then they would use a relative as a you know, second choice, you know, mom or a couple relatives, mom and the brothers, because of the maternal inheritance. So should we review that? Well,
that's what I'm basically getting at is what did they use to match this mtDNA sample they retrieved? How did they determine it was Teresa?
Well, they used Karen.
Okay.
So that what they determined was that the bone, the remain, the charred remain, and then this is getting off onto a whole other topic, because a mitochondrial DNA lab would always use bone. They would always use bone because bone is the Bone is, the tissue of choice. It's most protected. It's going to have the best tissue. So I don't know what they use because the report is so sketchy. Charred remains. Did they test bone? They talk about bone in all the other tests, but what is a mystery to me, and I have not gotten an answer yet, I'm not saying it's not possible, is that how did they get a viable DNA from burnt, grungy tissue and not from any of the bone fragments, which would be, bone is a much more protected tissue. I don't know. Anyway, that's a question I have. I have a lot of questions. So anyway, and plus we don't really know. I don't really know what they tested, but so technically, the way you would interpret this report is that the remains are consistent with Karen Hallbach's buccal swab, the mtDNA. Now, by default, you can say, since Teresa Hallbach should have the same mitochondrial DNA genome as her mother, that it was Teresa Hallbach. But again, as a purist, it bothers me. Then why didn't they take a sample of? And I know they're on a budget, I guess. I don't know. Maybe this is just how the FBI does things. But why didn't they take a sample of something from Teresa Hallbach? That would certainly confirm it, and compare. They never compared the remain against something from Teresa Hallbach. Now, some people would say that might just be their protocol, that's good enough, that we can just assume that since it was, her mother, it should be the same profile. But it also could be used as an argument. Do you know what I'm doing? I'm not saying one thing is right or wrong. I'm just saying if I was playing attorney, which Travis can laugh, which I do.
Travis had to skedaddle, unfortunately.
Oh, well,
he had something to go do.
Then that's something I would argue, and that was one of the reasons that, in my pictures, that was one of the arguments that why Buting had it thrown out. There were lots of reasons. What did he call the mitochondrial DNA reports? Cryptic and hard to figure out. So yes, that is, they compared. So your mitochondrial DNA now, as Lloyd said, in your nuclear DNA, you get one copy, of the chromosome, one copy from mom, one copy from dad. So you're 50%. Your mitochondrial DNA, except for weird exceptions, which we won't talk about because if it's the egg, it's maternally inherited. So a lot of these people, it follows a maternal lineage. Now, so a lot of people get confused, and the law enforcement officers did during this case, which was interesting because there's that interview they did with where, I don't know if it was Fassbender or Uyghur, went to Karen and asked her if she had any dead female relatives. And people often ask me why. I say because he didn't understand the mitochondrial DNA. Because people hear maternal and they think that it's, women. But okay, in genetics, circles are women and squares are men. So this is the mom and the dad. They have the kids. So the mom's mitochondrial DNA is passed on to all the children. So they all have, so if these are males, so she has these kids, they're all going to have. So Teresa's siblings all would share the same mitochondrial DNA. So Mike Hallbach, okay, but it's just that Mike Hallbach is not going to pass it on to his children, whereas the female children will pass it on to their children. Right. And so the same thing now with Karen Hallbach. So she'll have all of Karen Hallbach's siblings, will share the same DNA, but only her sisters will have passed that on to their children. So Teresa would have some cousins that would share the mitochondrial genome and some that wouldn't. So that's how that works. But it's passed on, and it's thought to be because there's so many more mitochondria in the egg, and the sperm head comes in and it falls off, so it doesn't get in there. But it's in general, it's maternally in here. So it's a straight maternal line, and that's how they make that. That's how they made that, assumption. It can be, that it could be assumed, and and it is a fairly reasonable assumption that if the remain, I say because they tested one thing as far matched Karen Hallbach, that you could by default say that could likely belong to Teresa. But again, and then of course there's statistics involved in all that business with science, but certainly no one is ever going to, science, only the press and the media and Pagel and Kretz will use the words confirm and absolutely.
How similar can Carmen Boutwell's mtDNA be to Teresa and Karen?
Yeah. Oh, hi, Tony. Well,
I'm actually the one I'm talking about is from Lindsay.
Yeah, I was going to answer Tony's question because they I don't I don't. Again, you in that popular. I know the Carmen Boutwell thing comes up a lot. There's no maternal linkage that I've been able to find tracing their family trees all the way back. So there's no reason to say they would share the same mitochondrial DNA profile. How similar profiles are in that region of the country, I don't know, but there's no indication that they are related. And Tony's making a comment about they used muscle tissue from the charred bone for the DNA Culhane. She does report that. However, and the mitochondrial DNA FBI analysis, I guess I'm not sure what they did at the FBI, because in the report it does not say. It says that they received a piece of charred remains, and I guess you could assume that they used the muscle tissue. And I do make that assumption, I guess, but I can't know for sure. I can't know for sure because it's not written anywhere in black and white what they used. So, but that's what I would assume. Yes, but I don't know. And was it muscle? I don't know. Or was it tendon? You can't really, is it tendon, muscle? I mean, it's just, when you look at the picture of that bone, it's not like, I think a lot of times people think about like they're seeing like steak, like meat on a bone. It's very fine. It's like the grizzly stuff, like looking that you see like on a bone that's cooked, that they scraped off. So is it muscle? Is it tendon? Is it, that's why they keep calling it tissue. I don't even know if it's muscle per se. I honestly don't know. Right. That's why I'm not saying it's not. I'm saying I would not say it is, because I can't say I absolutely know. And the FBI, I don't really know what they did. When you call something a charred remains, and then they're calling charred remains, bone plus. Sherry initially called BZ bone plus tissue, and then she sliced tissue off, and then that became BZ. And the profile BZ is from this tissue. But BZ at one time was referred to, apparently, a piece of bone plus tissue. Do you understand what I'm saying? I mean, these are subtle technicalities.
Yeah, subtle differences.
But BZ is, people ask me, what is BZ? And I say, well, it depends on when you're referring to it, because apparently when it came in as evidence, the bone was still on the muscle. Or BZ was actually two separate pieces of bone plus tissue, I assume, because she cut it off. So yes, Sherry tested tissue. What the FBI, because I would assume they would say bone if they did, but I honestly don't know, because in their report they don't say. So you can assume, but you can't know. And so I'm always really careful in this to not, unless I know for sure, unless I see somebody's report, which may or may not be accurate. But I mean, assuming it's accurate in black and white.
Lloyd Bass says BZ is BS mistyped. Another typo. What do you
think? Well, Lloyd is a good one with the.
He's just well. That's what he said anyway. So, how could soft tissue survive the fire? I've burnt ribs in my oven and they couldn't even be eaten. Never mind tested.
Well, that's a question I have, and I have not got a good answer from it yet. why did this one piece, and this is again where we go back, and I don't know how many people
It would seem to suggest maybe that
read
whoever
my
was doing this was keeping one piece a little less cooked for identification purposes, maybe.
well, that's what, I mean, we're getting, kind of gross, but I don't know how many people read my Reddit post, but Dr. Eisenberg went in the trial. In the trial, they made it very. They made a point of giving the perception that the bones that were tested came from, Stephen Avery's pit under tag number 8318, and she talks about this one. I call it the magic bone. All of these bones in her testimony were, calcined and burned, to the point of almost being calcined, except this one piece. And she shows that one bone in the left. I don't know if you want to put that picture up, or if you don't want to bother. That's picture number 4. She shows these bones, and there's the one in the corner that to the naked eye doesn't look any different than the other ones, but she claims that this bone is unburned. Yes, that one over there in the left. She says that bone, that largest piece, is unburned, basically, or not unburned. It appears as in a non-burned state. And somehow it was protected by this tissue. Well, I don't know. I mean, I'm no forensic anthropologist, and I realize, you can't be careful. Pictures, don't always, show what you're trying to show. But you can see it doesn't look, that dramatically different from the other parts.
No, certainly doesn't.
Anyway, right, Tony, there's no mention of bone testing. So I agree. It probably, I think they would say bone if they did test bone, Tony, but that behooves the question that this person brings up. Why then is there this one piece that was so different than all of the others? And how could it come now, and then I know, reading the literature, sometimes, bones are burned unevenly in a fire, but this one was very different. I mean, to have tissue, well, if it's sunburned, I'd hate to see it burned. Yeah, when we're talking tissue, there's like tiny pieces of tissue on there, and it only takes a little bit. Now go up to show number 5. That's SC's bone. You see what SC did was just basically crop out. She just cropped out that picture because you see the little bones around it are
turned it sideways.
And turned it to make it look like she used the same bone. Although, she doesn't say it, which again, I think they did a lot of things to give the perception
right.
that
that's clearly the same picture.
Right. To give the perception that the two were the same. However, Eisenberg testifies she did not send that bone to the crime lab, a la Sherry Culhane, that she packaged it up and sent it directly to the FBI, which we also know now is not true. So anyway, that's purportedly the bone Sherry says she cut her tissue from.
Right.
So.
All right.
Yes, BZ was in the original trial testimony I mentioned multiple times. It was a complete fluke, any tissue. Right. So exactly, that is an excellent question. And why did this one piece, and Ken Kratz mentions that, in his, number two is kind of nice, kind of little verbiage from Ken, where he dramatically in his opening statement talks about how, and I don't know where he gets the shin from, honestly, because Eisenberg never calls it a shin in her testimony. And then the state called it a femur. I'm sorry, nobody else.
Ken loves creative writing. Just read his book. Anyway, sorry.
But I thought that was, probably nobody else did, but I thought it was hilarious. They don't even know what freaking bone there. They have femur, shin, it's all in the leg. So, but Ken states that, it was a tibia, but how? Right, exactly. And I can't answer that, and I don't know. I've been trying to research, are there other cases where You know, they've had like cremated bodies, and you've got this one piece of tissue. And then, you know, Pevito talks about like, you know, these golf ball pieces of tissue, you know, sitting around on the ground. I mean, that's just bizarre. That's just bizarre. So it makes no sense. So I don't know. One thought, and I don't mean to, you know, go off on a tangent, because is BZ, there's no tag number for BZ. There's no chain of command for BZ. Fassbender made a phone call to Sherry on the 11th, the same day BZ was taken into the lab, she said, and said, he said, try to get DNA from the tissue. So Eisenberg was just examining these bones on the 10th, I mean, into the night. So how would Fassbender know anything about what was the deal with those bones? How would he know to tell Sherry to get tissue? You know, this is speculation only, but I wonder if that bone wasn't given to Sherry on the sly. I mean, somebody, Fassbender, knew where that bone came from and knew it would be able to identify, or possibly identify Teresa Halbach. And again, that's speculation, but but did that bone come? It clearly came from. I shouldn't say clearly. It would be a reasonable hypothesis to say that bone came from a different place than the other calcined bones.
Right.
To suddenly, well, what did it do? Fly out of the fire? She was supposedly in these Daisy Fuentes genes, and you know when it is kind of morbid, but there's a Dr. Elaine Pope who's supposed to be, I think, is the bone expert. Right, Tony Kretz doesn't want to go to the quarry, and we all know why. But she is the bone burning. I mean, she works out of this place in Tennessee. She actually burns bodies. You know, she gets cadavers and she burns them under all kinds of. She is like the burnt bone expert, and I've actually written to her, but she hasn't written back to me. although I think once people get the gist you're talking about this case, they don't want to talk about it. So I think I was a little too overt. But one thing that I did read that she wrote is that the first thing that melts is the fat. And so if she's in these genes, you've got, and I'm talking about, you know, the story, you know, according to the story, which is, this is probably not how things happen, but this fat would be burning and would have encased her legs almost like a sausage casing. So how did this fragment of tibia fly out of the fire to be in a relatively unburned state? You know, that does not make much logical sense. It's another Manitowoc miracle. So, you know, I mean, I think with the cuts and stuff, probably, I mean, again, it's kind of morbid, but the body was probably dismembered and then this piece. Yeah. Anyway, you're yawning, and this is a lot of
sorry,
science.
No, that's okay though. I'm sorry, I can't help it, but we're fine.
Yeah. Well, it's heavy stuff. I know this is heavy stuff. So.
No, it's not. It's seriously not that.
But I'm glad.
It's seriously, Erekose is two people burning the candle at both ends, and that's why. But anyway.
Yeah. Well, no. It's, you know, we try to liven up the science and make it a little exciting. So maybe it's getting but hopefully that clarified. I don't know, did the STR and the just even though I didn't go into any, I mean, we could even have a whole, I didn't really go into it at all, but just the basic differences between the STR and the mitochondrial testing.
I think we've covered that.
Did that help? Did that help people?
You guys feel like that helped you?
I mean, some people already understand that, but to the people that didn't, was that?
I think we went through it pretty well. For those that didn't understand it before, I think they may have gotten at least a better understanding of it.
It was a little. I mean, again, it was very basic, but I wanted people to just, because that's why I asked the question, because, you know, sometimes you throw out these, and like Ziggy said, that's a good point. No, you're not stupid. You just, you know, how would you know what these things mean if you didn't happen to study it in school? So people shouldn't feel. 4 a.m. I want more, please. Oh, there'll be plenty more. So, should we just so that was a lot. That was a lot of information. There's so many tangents we could go off on now. So should we just stop
If
and
you
take
want,
questions?
we can leave the next science segment for next time, and right now we can just talk about the ridiculous response from the
Let's
state
have
and
some
have some fun doing that.
I think so.
All
Yeah,
right.
okay, because that's enough deep for now. Yeah, I think it's no, seriously. I do think it's you can only, what do they say, after 40 minutes you're going to start zoning out. You can only take so
Yeah,
much deep
so much of
thinking
that.
at
Yeah,
once.
I
So
suppose. Yeah,
it's
probably
better
better.
to break.
We'll
It's
guarantee
better
in
to
small
break.
doses, folks. No, we'll get
Yes.
back to the fun stuff.
Okay.
The states had basically admitted that they gave the Hallbox animal bones. Let's talk about that, folks, because that's really the thing that needs to be. I mean, that's the thing that needs to be front and center, dude. Is people talking about, wait, what's going on here? Did the state just say
Exactly. I
did the
know.
state just say they gave the victim's family bones they don't know what they are?
I mean, how tacky is that? They're so concerned about I just about when I read. First of all, oh, we could go on. When I read the state's, what is it, response, reply? I don't know what you call it. I was just floored that they would actually have the and okay, living in Wisconsin, what do we hear the mantra all the time is, oh, the poor Hallbox, oh, this horrible attorney making the Hallbox go through all this grief and question,
Right.
you question anything, it's the poor Hallbox. What have they
Poor
done?
Hallbox. Yes.
Basically, they admit in this legal document, oh, we just chucked them a bunch of bones. We don't really know if they're human, if they're animal. And that's what I couldn't believe they actually did that. I would think they would have tried to fake that or something.
And the proof that they knew that they were wrong is the fact that they were holding back all the proof that would have shown Stephen's lawyers and anybody else what they'd done. They were holding all that back. I mean, it's like, dude, can it be any more obvious? You did wrong. You knew you were doing wrong. You tried to hide it. Oh my God. It's just, they are so dead to rights on this one. If the courts allow them to wiggle out of this one, I'm going to be super pissed.
Oh, yeah. and I know the fact that they even tried to justify, this, and they clearly broke the law. I know it's but again, well, okay, I'm not, I don't know the law, but to me, you can't. Okay, you've got all these different bones in let's say, for clarity, or 4 different places. Well, you identify a piece from one place, and I know because we talked about this before. They supposedly only found bones from one body, but you really, again, from a scientific perspective, you can't assume just because in one place they're all the same that means that they're the same in all the other places. So in other words, I would you would need, just because this piece from this other pit belonged to the same Teresa Hallbox, doesn't necessarily mean this other collection did. I mean, it's not like you're just looking at one body, or one even collection of bones. They're all, the possibility exists, and some of the fragments are so small, and forensic anthropology is a bit of an art, in my opinion, as much of a science that I know you can't, without DNA always say. And the other thing that Zellner's expert, not to go off on another, we're ripping on her. The other expert did bring up that she didn't even do, without DNA, what would be considered the rudimentary. Oh, we didn't even get to my particles either. Oh my gosh.
Yeah, we'll get it next time. No worries.
Oh, the particles.
Remember, we're not going to overload everybody. You want them to pick it up, right?
Oh, yeah, that's right. The particles. That's big. Okay.
But
yes.
But for the next episode, we'll do a brief recap of the stuff we talked about today, just to kind of bring everybody back up to speed a little bit, and then we'll go into the rest of your presentation. So it'd be good to go.
Yeah. Well, I knew it would be too much for it. There's just too much. Oh, but the particles. Oh, that is fabulous. That is fabulous.
Well, something to look forward to next time.
So it'll be interesting. So what happens now? How much time do they? It'll be interesting. How are they going to even come back from this?
Oh, I know, it's ridiculous. What are they going to end up saying, right? I want to answer one thing here, Ela. I'm not sure how to pronounce that, but They determined that the hood latch swab was not blood, urine, semen, or saliva, and that basically leaves skin cells. So that's the best answer we get on what type of DNA it is. So I hope that answers your question. Bones are right in the yard in a box and left the yard in a box. A lot of people think so. I kind of think that may be true as well.
Well, we don't know. You're welcome, Ducky. Good to see you here.
Hey, Ducky. Can Judge Suck at this cognitively tell the difference between animal bones and human bones?
Well, that's the other thing. And then the people picking the bones out, were what? Phelan and Gahn? I mean, were the ones deciding which bones to give back to the hull box? I mean, would they know? Like look at what those fragments look like. Would they know? It looks like the garden mulch, that you see outside. I mean, would they know like what's a bone and what's not? It wasn't like they didn't even have the anthropologists pick out the bones to give to the family. It was like, well, okay, well, this looks like a femur.
Right.
I mean, it's funny, but it's not. I mean, we can use humor because humor is useful. Lois is falling asleep, laughing.
Sometimes you either have to laugh or you scream or you'll go crazy.
Right.
It's documented from the state using non-human bones. Yeah. So yeah, that's the bizarreness there. And then in Zellner's, reply, she points out just a lot of the disingenuous things they were doing. Like, first of all, she starts out with the fact that it's not actually a reply; it's a motion for leave to reply. Okay, what she's doing is she is basically showing respect for Judge Squitz and basically saying, look, the state just went cowboy on you and just filed a response that was never asked for or solicited, but we've read it. We have concerns about it, and so if you do read it, we would like to submit this reply to it to answer some of the misstatements and disingenuous things, I guess, in the state's response. And so it's basically a motion asking for permission to reply, which will only really matter if she does read the state's response. But it's so funny because in it, she's very keenly pointing out the state just went cowboy. She didn't use the word cowboy, but that's what they did. They just went on their own and filed a response without being asked. And so she's now going through the proper way to do it, trying to show respect for the court, and that sort of thing. And whereas the state is now the ones making up weird stories, saying very strange things. And, so I think she's now trying to she's just trying to go, what, we're just going to respect everything and try to be as good as possible here because the state's losing their mind. They're going off the rails. That's what it looks like. I mean, seriously, I don't know how Gahn and Fallon got. Gahn wrote the statute.
Oh, did he? I was wondering who wrote that.
Yeah. Gahn was the one that wrote it. And then he and Fallon were both there that day handing these damn things back. It's like.
Well, I assumed he used Ken Kratz's book as a reference because Ken, in his book, talks. Ken, who made, in his opening statement was all about the shin. He talked on and on and on about Karen Hallbox, and this is about your daughter's shin bone, identified her. And then in his book, he wrote about a femur. And now it's a femur in the thing. They don't even know. They don't even know their bones.
So there was that. That was interesting. There was she goes into explaining that they're kind of dismissal of Andy rapid DNA testing. She talks about how they're relying on very old cases and literature and stuff for the information that they put in their response. She points out that they're ignoring the recent, the past 3 years where Andy rapid DNA has been applied on criminal investigations. A couple states have made it the way that they do their DNA testing in their state crime labs and stuff. It's starting to catch on, and and the state was
their
completely
response
disingenuous. Well, the state was completely disingenuous, basically saying that it was not getting wider use. It was basically not, none of those things were happening, and that was just a disingenuous statement.
They didn't understand what they were talking about. They
Are
were
you
they were
making excuses for them?
Yeah, they didn't even. Yeah, that was there. Like I told you, I would have loved to see. Richard Selden did such a great job in his response with like actual, studies and case reports, and I would have loved to see his response to his first draft or whatever. His response when he read that, he was probably like, "What the?"
Mm-hmm.
Because basically the Rapid DNA Act, all that does, it has nothing to do. The Rapid DNA Act does not say, scientists can't use this technology. It's just the technology. All that did was, when that's a whole separate topic, all that did was actually give law enforcement, was expand the use of it, give law enforcement the ability to now,
Right.
test your DNA out in the field, and they want to use it for homeland security, for all of that, for your protection, which is should always make you worry. So that's the flip side of it. I would feel much better with this being used by people like scientists who know what they're doing.
I'd feel better about that too.
Joe, law enforcement, but that's another topic for another channel for another time. But they misinterpreted the meaning of the act, and like she said, that was just it was. I'm not a legal scholar, but that was pretty bad what they wrote.
Judge Suckett wouldn't have read it, so he can still do so at COA. I heard Wiegert is going to be the new sheriff. He is the new sheriff.
He is the sheriff.
He is the sheriff of Calumet County.
Yeah.
There's no gonna be. He barely won. Barely. Literally. By the skin of his teeth.
I know, isn't that scary? He's a sheriff. Well, all of these people have get promoted.
But he wasn't sheriff when Kathleen Zellner's investigator was petitioning Calumet County with a FOIA request. It was actually Mark Ott at the time. And does everybody find that funny? That the letter that the state is saying, where they told Mr. Kirby that they sent the 1,117 pages and the additional 60-something pages of the post MPCR investigation, like that letter that says both of those were sent over, but that one is the only one out of all the letters that Kirby has. That is the only letter that was only shown to him because it was in the state's filing, their response. So it was showed to him. He doesn't have a copy of it, but he also notices that it's not signed and it doesn't have the same stamps as every other piece of correspondence that he has from Calumet County. That is more than a little bit interesting to me, because that was basically attached in the state's filing, and he says that it looks like it came out of a 3-ring binder. So very interesting, obviously.
So lots of people aren't here tonight, but there's only 20 likes. We need a T1 to say smash the like button.
Smash!
Science doesn't always draw a crowd, but
I got a thumb down.
For those hardy people here. But it's a forensics case. Understanding the sciences is key.
They do owe Kirby some money. Where's my investigation report? This is all just going to come back and bite them. There's so many ways now that you look at this where the state was hiding things, hiding the CD, hiding, the way that they unfurled the whole computer evidence and all that sort of stuff before trial, messing with Dean and Jerry so that they wouldn't pick up on the content of that computer and stuff. Look at the way that they're doing with these bones, giving them back, not telling anybody they did it, handing them over, not telling anybody, not handing over those ledgers, not handing over those pages of the investigation report, basically hiding what they were doing, knowing that they did wrong. Right? I mean, this is what it shows, and it's important to talk about and to see and to notice. They knew they were doing wrong, and yet they just did it anyway. And the reason why you can say they knew they were doing wrong is because they were trying to hide it.
Yeah, and I know you said people might not be as excited, but again, and then I go into it in the Reddit post, but in my opinion, they intentionally deceived people into thinking that bones, that Leslie Eisenberg wouldn't forget what she sent to the FBI. They intentionally, again, it's just the level of deception, like Zellner tweeted. They intentionally deceived people into thinking what was tested came from Stephen Avery's burn pit and was that bone in that picture that Eisenberg showed that was the unburned bone. Oh, come on. And that Sherry, that was Sherry used the same bone in the picture, and that, she got that bone. And then people just assumed that these things that were tested came from Stephen Avery's burn pit, from this one batch of bones that were also and we never even got into we have so many fun things to talk about. And because those were the only bones that were used in the autopsy, which is the legal document that makes Teresa Halbach dead. And as the physician that performed the autopsy said, the identification was dependent on the DNA. And since the only bones used were the ones collected from the 8th to the 10th of November, it was necessary that the identification came from something from those pieces. If someone was ever to dig into things, which fortunately for them, probably no one ever did. But
do I have a working queso link? Yeah, I do. Hold on. Wait, not on this tab. Oops. Go to. Bear with me a second.
Oh yeah, Lloyd, that was that was the Dr. Jensen, the medical examiner that got to testify at the trial to state a cause of death. He was the one that said there was blood on the bullet, and that's how he knew Teresa was alive when she was shot, which is another appalling. Apparently, he left his Hippocratic oath and long at home.
It's more like a hypocritic oath, right? No.
Yeah. When he made that statement, because no ethical MD would claim to know a cause of death, and then he didn't even read the lab report. Oh, that was so bad. We could go on and on, but. Yeah.
So I want to talk about one other thing about this reply. Obviously, guys, I'm going to be going over it with Tracy and Travis on Monday. I'll probably be talking about it all weekend just because how great it is. That reply was awesome. I mean, I stayed up till midnight, even though I had to go to work today. I stayed up till midnight reading that thing and posted on Twitter because, oh my God, man, she is really, I mean, it's like she's just got the state up on the ropes and she's just like, it's like awesome. Anyway. And so the next thing I want to talk about is how Zellner and her associates must have been sitting down at the table getting ready to write that brief. And they're like, oh, okay, yeah, so first thing we're going to do, we're going to ask for a motion to leave to file this. And we're going to point out exactly how much the state just went off on their own, did what they did without being asked, but we're going to ask for a motion to leave, whatever. And then they started going in, they probably talked about what they were going to say. And then they got to the point where, I know, I have a great idea. I know what we'll do. We'll use this particular case, and I can't remember the name of it right now, and we'll argue that the state is procedurally barred from arguing that we are procedurally barred from bringing this claim. It's like somebody was like sitting there going, let's take the state's argument and turn it around on them. I was having so much fun reading that reply last night. That was brilliant. Absolutely brilliant.
Yeah, Russell, it's winding
See you
down.
later, Russell.
Thank you for being here. People are getting tired. Yeah. And the other, yeah, it was.
I don't know that it'll ever be investigated outside of Wisconsin other than by us web sleuths. But I doubt you're going to get an outside agency, Russell, to come into Wisconsin. Hopefully, maybe a federal one, but geez, if that hasn't happened by now, you got to wonder.
Well, it would be nice, you know, because I've compare this a lot to the Amanda Knox case because there's a lot of similarities with the DNA. And what they were able to do in the Italian system, what the Italian Supreme Court did was they just asked for some independent scientists to go in and take over, basically, and look at the DNA. And oh my gosh, they found all kinds of problems, and we tested it. And I mean, ideally, that would be nice if that could happen here, you know, but it won't because of the way our system works. You know, just somebody completely independent and private could just come in and take all the stuff and just, you know, a good forensic anthropologist, a good, you know, someone that you could trust.
Right. Yeah.
And a good DNA person. But I guess that's not the way our system works.
Yeah. All right. Well, I think maybe it is getting a little late for everybody.
Yeah.
It does seem to have slowed down in the chat there. So unless anybody has any other real quick questions, I'll take the next 2 or 3 if you got them before we go. And we're going to go out listening to Stacy, by the way, just so everybody knows. But 2 to 3 more, depending how quick they come in. All right.
Okay.
That's not a question, Lloyd.
Let's see. Scooby the dog. Scooby the dog.
Where's Scooby there?
Scooby dog says good night.
Hey there, Scoobs.
Is somebody showing him?
Oh, where'd he go? Oh.
There. Can you see him?
No, he's just off the frame. There he is. Okay, I see him up the corner.
Yeah, he's gone. He's gone to bed. He said good night.
All right. When is the topic of rapid DNA going to be discussed? Well, we talked about it a little bit tonight, but you know, we can make that next time. We talked about it though. We didn't talk about it tonight. We'll talk about it a little more in the future, Russell. You're welcome, Charla. Why did Sherry Culhane use all the bullet sample when she did the PCR test when no one ever uses all the sample? Well, I think she felt that was her only hope of getting enough DNA because it clearly wasn't visible DNA.
Yeah.
That's my best guess at that. So.
Yeah, I mean, there was nothing visible and it was such a small piece. The only, you know, that would be the logical, you would just dunk it in the tube of buffer. Yeah, there was nothing to swab. That was her only hope of extracting any.
You're all very welcome.
Although it was very bad judgment to be training. What was her story? Training your trainees and talking over it, spewing
we could have
out
trainees there, but we couldn't have defense and their experts or
during this critical testing where you knew you were going to be using up your sample. So that, you know, just kind of goes to the judgment there. But yeah, because talking, we know that we send out aerosolized, which Joe doesn't say exists, right, Lloyd? Aerosolized cells.
Saliva cells?
Saliva cells, which contain...
Oh, the genetic code. DNA.
White blood cells.
Yes. Right, which have
I know, we're getting close. Right. See, I'm making you guys think it's too late. It's too late. Even I'm getting tired. Yeah. Tony, I know. Yeah, we could. Oh my gosh. Yeah. Talk about bad lab technique.
All right. So everybody remember MAM weekend, MAM-a-thon coming up. Always, you know, keep that in mind. We got that coming up. Something to be excited about, something to think about. You know, try to tune in that weekend, get into the live chats, try to maybe visit multiple YouTubers. Show us all your support. Obviously, Tracy and Paul and Allie, get on their live chats and encourage them. And because Allie and Paul and Tracy aren't really as accustomed to going a lot. Well, Allie's getting there. Allie's getting pretty veterano at this point. But Tracy and Paul aren't so much. So show up and give them some support, or try to make a point to, at least to help them. Encourage them. Anyway, we're gonna go out listening to Stacy. Anything you want to say before we go into Stacy there, Scoobs?
No, good night. Thank you for having me, and thank you for being here, everyone. And great questions and participation.
Yeah, and thank you for being here. It's been my pleasure to have you on.
Always. Okay, let's rock out to Stacy.
Here we go.
Now we have some. Always. Okay, let's rock out to Stacy. Here we go.
So let's keep talking about Avery. Yeah, let's keep talking till he's set free. Oh, you just listen, I think you'll see why it matters. Yeah, let's keep talking, really matters. You just listen, I think you'll see why it matters. Put aside the documentary. It's personal mediocrity. Oh, all their claims so blatantly get torn to tatters.
We're getting some audio on your end too.
I think that matters. I bet you heard about Hood Maxwell, but maybe you ain't heard this yet. 19 more times the DNA that was left anywhere else that day. I think that matters. I really do. I think it matters. An ex-boyfriend with a day planner page, and yet they did not investigate. And who knows why, and who can say why he was signed into the crime scene that day. I think that matters. I really do. I think it matters. About the barrel that once was seized, but then returned to the Averys. The bones were found the next day, but not one photograph exists of where they lay. I think that matters. I really do. I think it matters. I could go on listing things off. 80 more things.
Finn!
I just keep on singing these songs till Avery freedom reigns. I said it matters. It really, I know it matters. It could be you, I think it matters. It could be me, I know it matters. Work with Casey, it matters. I said it matters. It really matters.
All right, folks. That's it for today. Look forward to MAM-a-thon. Tell everybody about it that you can think might have an interest in MAM. Maybe they used to be, and or something like that. Anybody you think might have, because some people lose interest in stuff. So let people know it's something interesting they might want to check out if they ever had an interest in MAM. Whatever it takes, let's create as much interest as possible, and and do that thing, right? I mean, we want this to be successful. Obviously, we want this to go well. So, keep talking about it. Just like everything else here, keep the conversation going. Start the conversation. You guys know I keep pounding on these hallmarks. They're important to me. That's why I keep pounding on them. So that's about it for today. If you haven't already, please hit subscribe, and we'll see ya.